In this video, we're going to be looking at anion exchange chromatography. We set the column up in the retort stand and add a small amount of the 17.5 millimolar phosphate buffer. The reason we do this is to remove any air from the glass wall that is used to plug the bottom of the column. Using a glass rod, we gently tap the wall at the bottom until all the air has been released. Do not press the wall down really hard, as this will clog the column. Once we've removed all the air, we drain the buffer from the column until the meniscus is almost touching the top of the glass wall. This is done by adjusting the tap on the bottom of the column. We will see this tap later. Next, the DEAE-Sepharose is added to the column. Make sure the material is back into suspension before quickly pouring it into the column. Pour in one go, filling the column to the top. If you have excess column material, you can open the tap on the bottom, not too much, just a little bit, to allow the excess material to drain out. As the level of liquid falls in the column, you can then add in the remaining material. One key thing here is not to let the column to start to separate out too much, because you do not want to form a line between material added and material already poured in. This process is repeated until the column is full. Once all the material has been added, the column can be left to settle. Here we've speeded up the process. 7 minutes of column settling is running over the space of around 30 seconds. As you can see, the column material is settling and a clear layer of buffer is clearly visible above the bed of the column. We let the buffer drain down until the meniscus is almost touching the top of the column, before we top up the column with additional buffer. Here we're washing the column to return its condition to that at which it was originally in the tube, that is pH 6.5. We fill up the top of the column and we let the buffer drain through. We will do this a number of times until the material flowing out of the bottom of the column is at pH 6.5. This may take 2 or 3 washes. Again, the tap is opened to allow the liquid to flow from the column. We've adjusted the column so it's flowing at a rate now which we can use to collect fractions. As the meniscus comes down to the surface, the top of the column, we will then add in the protein solution. This is a mix of Catalase and hemoglobin. We carefully drop her in the 200 microliters using a P200 pipette, and we let that run in. We then add a small amount of the 17.5 millimolar buffer. Just add a small amount and let that run in. This way we keep the band nice and tight. If you fill the column up to the top at this point, the band will diffuse in the liquid and you'll end up with a broad band. As you can see here, by adding the buffer carefully at the beginning, I've produced a nice tight band of hemoglobin that can be seen moving down the column. Once the protein has entered the column, we can then fill up the gap above the column bed with buffer. The column must not be allowed to run dry at any time during this process, so it's always a good idea to keep a good amount of liquid on top of the column. As you can see, the protein is running down the column. It's just passing out of the bottom of the retort stand clamp here. You can see it clearly as the brown band. When the protein gets to this point, it's time to start collecting the sample. Taking one of the tubes from the rack, we place it underneath the tube at the bottom of the column and collect 10 drops into the tube. All the time keeping an eye on the buffer in the column to make sure that the column doesn't in fact dry out. Keep topping up the column even whilst collecting. As you can see now, the protein is in the process of leaving the column at the bottom. Here we can see the sample collection. We collect 10 drops into the tube, and we want the column to be dripping at a rate such that it is easy to swap over between tubes. You would have adjusted this drip rate when you were washing the column. It is vital that you have the tubes ready in the rack because, as you can see, you need to move quite quickly between samples. Here we can see the meniscus of liquid just touching the top of the column and the protein solution being loaded. It is critical that you do not fill the column with liquid at this point, because if you do, that protein band will disperse. You just add in a small amount to let that protein band move into the bed of the column. Here you can see it beginning to move into the bed of the column. Once the protein has entered the column, it is then okay to fill up the gap above the column with buffer. In fact, this is advisable as it means you're less likely to let the column run dry. The protein has now entered the column and we can add in more buffer until the column is full. We do this carefully as we do not wish to disturb the bed of the column too much. We do not wish to cause a massive disturbance in the top surface. Here we can see the hemoglobin band at the bottom of the column. It's just about to elute. It's moving into the glass wall that we used to pack the column earlier. It is critical that this stage is carefully watched, because ideally you wish to start collecting the protein before it leaves the column. Otherwise you're going to miss it. As you can see, the band is fading now as it slowly leaves the column and is collected in the tubes. Once we have collected all the hemoglobin and washed the column a couple of times, we run the meniscus down to the surface again and then add in, drop-wise, the 175 millimolar phosphate buffer. Again, we add in a few drops, let it run in, add in a few drops and let it run in. This way we keep the catalase band nice and tight. If we just filled up the top of the column with buffer, the protein would unbind from the column and diffuse into the excessive amount of buffer above it. This would cause a broad band to be eluted at the bottom of the column. Once we've added sufficient buffer and the protein has entered the column, we can then fill up the top of the column with buffer. Again, we must make sure that the column doesn't run dry. The catalase band can be quite faint. You can just see it in this image. It is therefore important that you watch carefully and start collecting samples at the appropriate time. Sometimes you can't even see the band and you have to guess when it will be eluted. This can be done by monitoring the amount of buffer that you've added to the top of the column. Here you can just about see the band diffusing as it enters the glass wall and is eluted from the column. This image shows the hemoglobin tubes. As you can clearly see, there is a difference in colour as the hemoglobin was eluted from the column. Once you've collected your fractions, you can test them with hydrogen peroxide. Here we're testing the second elution in the 175 millimolar sample. As you can see, the first tube didn't react. Tubes 2, 3 and 4 have reacted strongly, small reaction in the remaining tubes. The hemoglobin tubes would not have reacted at all. However, you would have seen a clear colour difference between the hemoglobin tubes when compared against a white background.